Welcome everybody to today's edition of the EV Journal Club. This will be our second joint edition with the Student Network on EVs. So you can find out more about SNEV or SNEV. We've been having a conversation about how to say it, and we might resolve that today. But you can find out more about them by looking at snevresearch.org. Is that right, .org?
Yeah.
Yes, okay, so SNEV Research. And so this is a student network. It's a way for students to find each other, to learn about protocols, to learn about what other people are doing. And there are also numerous online events that they host with graduate students, postdocs, industry. So keep a lookout for what they're doing. So I would like to now introduce Camille Trinidad, who's at the University of Kansas Medical Center, who will be moderating this event on behalf of SNEV today. So Camille, thank you so much for moderating.
So thank you very much, Ken, and thank you for introducing SNEV or SNEV. We haven't really figured that out yet, but later we will. So today we're lucky to have the authors of this paper that will be presented today. We have Mathilde Mathieu and Dr. Clotilde Théry. Mathilde is a fourth-year graduate student at the Institut Théry in Paris. She is on her last year of her PhD, and she is finishing up this paper that is going to be presented today. Her studies involve characterizing EVs from different subcellular origins using HeLa cells, and she'll be helping out with the discussions later. And then our presenter for today is Aaron Scott, and he's a third-year PhD student working at the School of Physiology and Pharmacology at the University of Bristol in the UK. He is working on heart repair and regeneration in zebrafish, more specifically looking at the potential functional roles of EVs in this process. With that, I hand the screen sharing to you, Aaron.
So hello everyone, and thank you, Camille, for the introduction. First of all, I'll just say it's been a real pleasure to dig into this paper. I'm often left quite envious when reading in vitro literature, wishing we had the same sort of techniques and approaches that are so readily available, and that we could use them more in our in vivo systems. But I guess there are always these kind of trade-offs in science. So whilst this is not my specific domain, as Camille mentioned, I work on EVs in heart regeneration using zebrafish. This kind of literature, I think, is it's the sort of literature that every researcher in the EV field is following with a lot of interest. And also, I was just going to quickly note, within the first paragraph of the introduction of this paper, I was actually converted from being somebody that used the term microvesicle to more of an exosome person. But this is taking a little bit of time for me to adjust to, so you might catch me using the terms interchangeably. So I will assume that as we're all predominantly EV researchers here today, so I won't introduce EVs and their well-known biogenesis pathways, nor the often quite arbitrary and loosely adhered to definitions we've all heard time and again. But that's not to say that we should disregard accurate definitions, as they're obviously really quite critical to meaningful science. And this article published last year from Ken and Clotilde laid out some key considerations, concluding with the recommendation that the term exosomes should only be used specifically for multivesicular body-derived EVs and not for those budding directly from the plasma membrane, so our ectosomes. So this meant most EV preparation methods that are widely used would leave you with a sample of what should more loosely be defined as small EVs. And this is all very important because it's assumed that EVs produced by such distinct biogenesis pathways will likely carry different cargo and possibly then distinct functions. But this is yet to be definitively demonstrated, as it's difficult, obviously, to reliably separate exosomes from ectosomes. So then there's this proteomics paper from the Théry lab that was published in 2016 that I think many of us, I think it was one of the first papers I came across when I started working on EVs for my master's project. So they went into detail looking at these classic exosome markers that have been used for decades, CD63, CD9, CD81, and use these to immunocapture these EV populations and then carry out proteome analysis, which suggested that CD63 might be best considered the exosome marker and CD9 and 81 more likely ectosome markers. But there was a bit of a hurdle to overcome because this paper, like for things to then to be moved forward. So in this paper, they were using primary human dendritic cells, which obviously are not particularly easy to, well, I would imagine are not particularly easy to come by, and perhaps more difficult to work with. So the work that we're looking at today takes the next steps forward by moving into more readily available and amenable HeLa cells. So I took this sentence at the top of the slide directly from the preprint, and I think it nicely sums up how central these cells are to science today. And it's obviously a very, very old, well, a fairly old book by now, published over 10 years ago, but I recently started reading The Immortal Life of Henrietta Lacks, and it's been a great read so far. And I think it's definitely a history worth remembering, and very close to home, I think, for, I guess, many of you are from the John Hopkins University here today. So let's get stuck into the paper. So first of all, we have a plot that I think will be familiar in one variation or another to many of us, where we see the presence of CD63 and CD9 in the cell lysate, absent within the 2K and 10K fractions, and then present within that classic 100K ultracentrifugation of these samples. And then I thought it was quite, well, I guess quite simple, but quite a neat idea that they then went further and did a 200K spin and realized that there was a sort of residual CD63 and CD9 to be found within that fraction. So I think for the majority, if not all of the experiments, subsequent experiments, they opted to exclude the 100K spin and just finish with the 200K spin. And I guess it's kind of important to note as well that the isolation procedure is literally just based on ultracentrifugation. So quite a sort of broad isolation method rather than any filtration or more complicated isolation procedures. So then we have this schematic describing the immunoprecipitation approach to pull EVs from the sample using CD63 and CD9, which shows by presence and absence in the pull down or the flow through that we have three populations of EVs defined as the CD63 positive, you can see it done in this little schematic here, CD9 positive, and CD63/CD9 double positive EVs. And this data suggests that the single positive EVs constitute roughly 50% of the double positive EVs considered by the protein quantity. So my immediate thought that I had when I got to this part of the figure was the possibility of aggregations, maybe quite a petty thing for me to think of so quickly, but I thought of the aggregations that could occur that might skew these results. But it's pleased to see there's this CRISPR knockout experiment in the supplementary material that nicely demonstrates that these are likely single EVs that are double positive. So then we can take a quick look at the endogenous expression of the two tetraspanins and see CD63 within internal compartments and CD9 primarily at the plasma membrane, but with some rare instances within intracellular compartments. But in steady state, there appears to be no co-localization. And so the idea is that a more dynamic approach was needed to determine where these double positive EVs might be coming from. So then we move on to figure 2, where they introduced this rush system, which is completely new to me, but allows a protein of interest, so CD63 and CD9 in our instances, to be retained within a specific subcellular compartment, so the endoplasmic reticulum for this paper. And this is achieved by using a hook protein specific to the compartment that you want to locate the construct in. And this is fused to streptavidin, which binds a streptavidin binding peptide that has been fused to the tetraspanin of interest. So critically, this is reversible. So through the addition of biotin, the streptavidin fusion hook protein is released and the tetraspanin is free to move along its usual trafficking routes. So the take home from all of that, I've tried to break that down and understand it as best I can. But so what's clear is that very precise control over the localization of the protein of interest is possible until it's released. And then, well, and of course, we've also got the fluorophore is involved here as well, which allows the subsequent movements of the protein to be tracked by live imaging. And it's also important, I guess, to note that the modifications have been made at this small intracellular loop of the tetraspanins, which has previously been demonstrated, I think in a couple of papers, to not interfere with the trafficking of the protein of interest. So here we are. As promised, the biotin is added at time 0, if you like, and the tetraspanins can be tracked from there. So I think the stills here are quite useful, and the white arrows that you can see show the co-localization at 30 and 58 minutes. But the point here is that the live imaging, and we can see in this video the real dynamics of this. And of course, this is where the quantification can come from, which is really quite powerful. And we see here that between 15 and 40 minutes after leaving the ER, both CD63 and CD9, well, they enter the Golgi, and then they separate, which you can see in the decreased Pearson's coefficient, so the co-localization measure. CD63 appearing to move into intracellular compartments, and CD9 more towards the cell periphery. But between 30 minutes and 1 hour, a fraction of both proteins can be found in shared internal compartments. So then we have some very nice immunogold labeling of CD9 indicated by these red arrows, and CD63 by these blue arrows. And there was some quantitative analysis of this data giving a relative labeling index, but I can't pretend to have completely grasped this in the time that I've had with the paper, but I will give you the key take-home messages anyway. So it showed that CD63 and CD9 were found within the same multivesicular bodies 1 hour after biotin addition. So you've obviously got that in the top panel there, the top left. And at 2 hours, there seems to be less in these compartments, but is still present. And then it's also on the right, we're looking at the plasma membrane, and CD63 seems to only partially and transiently be expressed here. So then using confocal microscopy, they were able to confirm the partial co-localization of CD63 and 9 in RAB7 positive late endosomes. And interestingly, definitely, well, very interesting to me, some minimal co-localization was also observed between CD63 and membrane tethered mCherry. So membrane tethered in this instance using a joint myristoylation and hermitylation, always struggle with those words, tags. And between 20 and 60 minutes post biotin addition, and unsurprisingly, so you can see that in the co-localization here again, and unsurprisingly, much more and steadily increasing co-localization between CD9 and that membrane label, which you can see in the video. And then again, you can see that in the co-localization there. And I found this supplementary figure particularly interesting as it complements a small set of literature, for example, from Alyssa Weaver's lab and Zandra Breakfield's lab, and some of my own work that has looked at membrane tethered fluorophores and their inclusion or labeling of different EV populations. So I would be really interested to hear if anyone, well, anyone anywhere, but particularly here in the Théry lab, taken this figure kind of a step further and looked at the inclusion or exclusion of membrane tethered fluorophores in ectosomes or exosomes. Okay, so while I was looking through the paper, I put together this quick schematic just to keep things clear in my head. So initially, Mathilde has identified 3 distinct EV populations, 2 singular positive and 1 double positive for the CD63 and CD9. So then these rush constructs have then been shown, then shown us that CD63 and CD9 leave the ER and reach the Golgi with similar kinetics. Then we see the separation and CD63 positive EVs forming within these larger endosomes. Then we see consistent expression of CD9 at the plasma membrane, but only transient and limited expression of CD63 at the plasma membrane before they are jointly located in smaller endosomal compartments. But of course, the live imaging that describes the dynamics is limited in resolution. And so we can't be sure if these small compartments contain a heterogeneous mix of single positive EVs, or if this is where our double positive EVs can reside, but possibly quite transient too. So that's this bit that I've just included here. So the authors then go on to ask the interesting question: if CD63 can be found at the plasma membrane, can it also be secreted into exosomes? And to answer this, they mutated CD63 by disrupt the lysosome targeting motif located on the cytosolic C-terminal tail. And this pair of immunofluorescent images shows the different distribution expression at a steady state. Then we see the expression of the mutated CD63 is more similar to CD9 than to the wild type CD63, which also added, I couldn't not add the videos, although the stills give you plenty of information to take home, especially combined with these co-localization graphs. I think everyone agree these videos are too pretty not to show. So you can clearly see that co-localization difference in the analysis there, and the mutated CD63 acting much more like the CD9. So I'm not sure how common this approach is with in vitro work, but to me at least this was a very neat idea, where they wanted to confirm that the different tetraspanins use the plasma membrane as an intermediate and look more precisely at these kinetics. So by antibody staining with obviously with no permeabilization, they can label the tetraspanins exposed on the outer leaflet of the cell using obviously a different fluorophore. And then this can be used to calculate the proportion of surface staining over total staining. And this can be assessed over time using flow cytometry, confirming transient CD63 expression at the plasma membrane, but the mutated CD63 behaving much more like the CD9 accumulating at the plasma membrane. So they also carried out this internalization assay, which was similar to the previous approach, but with the addition of an incubation step, followed by a stripping of any antibodies remaining at the surface using trypsin, and followed the same flow cytometry-based analysis, which revealed, so in the quantification, revealed a minor proportion of mutated CD63/CD9 being re-internalized versus the known re-internalization dynamics of CD63, which is almost total and quite rapid, I think. So this is obviously a key complementary experiment for the live imaging data. Whereas now, so we're kind of moving away from the intracellular trafficking dynamics and looking at the end result as far as we are primarily interested anyway. So the loading of the tetraspanins into EVs. And we see immediately that there is an increased loading of the mutated CD63 into small EVs, which you can see in the blocks on the right. And the blocks here are being, for the quantification, are being normalized to what should be a consistent CD9 expression. Then they look at the same thing here, but using the immunocapture method, and again, show that same difference. Okay, so then we move on to the use of the ATPase inhibitor baflomycin, A1, which inhibits the acidification of late endosomes and has been quite widely used. I've even tried it in my own work as well. And sometimes it's been said to increase the release of CD63 positive EVs, which Mathilde obviously confirms in this plot here. But you can clearly see the mutated CD63 and CD9 are not so influenced. So combined with the previous slide, this data shows that stability at the plasma membrane increased CD63 release in EVs, and also suggest more abundant release of CD9 and the mutated CD63 in EVs compared with the wild type CD63, which is obviously more stably expressed within multivesicular bodies. So this is then confirmed in figure D, where the tetraspanin's expression in the small EV fraction has been normalized to the cellular, or I guess, so the cell lysate expression of itself. So the take-home message being that in HeLa cells, at least, small EVs are more likely to be secreted from the plasma membrane than from multivesicular bodies, at least under steady state conditions. Okay, so then we dive into some, well, to me, looks like very comprehensive proteomic analysis to further validate the origin of the different EV populations and excitingly to identify markers that can be uniquely assigned as identifying either exosomes or ectosomes. So after having validated the Rush constructs, they opted to isolate the EVs using immunocapture via the GFP fluorophore. They took 2 time points, 2 hours post-biotin addition as an early secretion time point and 24 hours as a time routinely used to recover EVs from conditioned media. So clearly the use of the Rush constructs became super valuable here again, as the precise timing allowed the assessment of EVs secreted at defined time points, for example, allowing the avoidance of too much reuptake and recycling, playing into the final results of the data. Also, importantly, the same GFP antibody could be used for the isolation of both tetraspanins, which allowed for efficient retrieval and direct quantitative comparisons. So in the interest of time, I'll skip through quite a bit of this. There's obviously a lot of data here that is very interesting to different people in different ways, and I think it's going to be a massive resource for a lot of people. But critically, this did allow them to probe further into the subcellular origin of these 3 EV populations that they've identified by comparing their data to a comprehensive reference database with specific assignments to HeLa subcellular compartments, essentially confirming the findings earlier in the paper with, for example, CD9 bearing EVs coming from the plasma membrane, whereas CD63 and CD9 EVs coming from both the plasma membrane and lysosomes. So again, there are plenty of details in the paper on how they arrive at these results, but the major take home, which is schematized here, is that they've identified some new markers that discriminate exosomes from ectosomes. So, sorry, I didn't take that slide out. I ended up combining it together. So then they can take these markers that they've well validated and use, so in this instance, they've used the neutral sphingomyelinase inhibitor, GW4869, which again is very widely used, again, something that we've used in our lab, and baphlomycin again to assess the effect on secretion of these validated markers. So whilst GW4869 came with some words of caution, perhaps only minimally affecting a subset of syntenin 1 EVs, baphlomycin was sort of more successful in the goals that they had. So the authors suggest that through this analysis of baphlomycin treatment on EV secreting cells, you have a method for evaluating the association of a given EV marker to exosomes, where you witness an increase in secretion versus exosomes where you would see no or very little increase in secretion. Okay, so then I was just going to finish on some of the questions that I had. But obviously we'll open this up to everybody else to have a good discussion. We've got Mathilde and Clotilde are both here, so we should have a good discussion. So I guess the first couple of questions are grouped together, which might just be part of my naivety with this kind of field, but I was left wondering whether you could add the lysosome localization tag to CD9 and see if CD9 expression is significantly decreased in EV, so like a kind of chimeric version, and then whether you could conclude and use that tag. Again, this is where the naivety might come in, but to target membrane proteins synthetically to exosomes. And so the next group of questions are kind of more broad, and I'm more just wondering if you might speculate on the answers to these rather than have a very definite answer. So just wondering how the dynamics that you've explored might change in response to adverse stimuli or sort of hypoxia treatment, for example, starvation, these kinds of things. How the dynamics might change within more complex environments. Of course, I'm interested in vivo, but that's obviously potentially a very big question. But even in potentially changing the culture conditions, popularly looking at 3D cultures or the inclusion of ECM components. And then I guess sort of summing that up, like basically I'll be interested to know where strategically you think the next important steps are to go from HeLa cells. And then I just have one that was of particular interest to me. So obviously you've divided the contributions of exosomes and ectosomes to the popularly isolated small extracellular vesicles. I'm kind of interested in the question of apoptotic bodies, just kind of because I think my impression is that it's always been quite an arbitrary division that we've got in the literature based on sort of size and density. And apoptotic bodies are often thought to be excluded by sort of lower centrifugation speeds. But in my mind, I can only imagine that apoptotic bodies, there isn't that limitation on their size, and they certainly could creep into the small EV fraction. I don't know if that's right or wrong. But also I think it's interesting because to a small degree in vivo, apoptosis will be occurring even as part of like the organism's homeostasis. So it's kind of very physiologically relevant. So I just started to imagine whether there are experiments that could be done that would allow you to sort of maybe induce apoptosis in a small proportion of your cell culture cells, and try to look at the same sort of dynamics and whether you can divide those, and maybe even answering the question that to me still seems unanswered of whether apoptotic bodies are the only ones that externalize phosphatidylserine, or is that something that some papers say is done when, say, by not microvesicles, exosomes. But yeah, that's all I've got. Thank you all for listening.
Thank you, Aaron, for that clear and wonderful presentation. So I don't see any questions in the chat box, but obviously here we can see that Aaron has a lot of questions for Mathilde and Clotilde. So you guys can go ahead and unmute yourselves if you want to answer some of these questions from Aaron.
Yes, okay. So thanks, Aaron, for the nice presentation. So yes, maybe I can start doing this. So for the about the to put the lysosome location tag to CD9. So I know it has been done to see if this tag at the end, at this extremity of the tetracycline is sufficient to target it to endosomes, and CD9 with the lysosome targeting motif targeted to endosomes. And then about the effect that it has on secretion in EVs, I don't think it has been done. Clotilde, maybe you can correct me if I'm wrong. So it will be for sure something interesting to see just if by being in an endosomes sufficient for a tetraspanin specifically to be secreted in exosomes. And then add this protein to load it into exosome. I don't know if it would work. I don't know. Probably not all things that are within an endosome are secreted into an exosome. So it would be interesting to see and to compare what kind of signal you need on the protein to be loaded in exosomes. But it's something quite difficult to precise. It's more complex than that, I guess. So I don't know if anyone has a comment on another question of this on this part first, maybe. If not, to the other questions.
So maybe I just can add something, Mathilde.
Yeah.
Sorry. Well, in fact, Steve Gould has done something, has used also some mutant of the C-terminal, the C-terminal part of CD9 and CD63. So he had a bioarchive paper a while ago, and I remember before he had the paper, I had met him at a meeting, and we had the same results as him because Mathilde already had this CD63 mutant results there, and so we had the same results. We did not do the CD63 tag to the CD9, but he did it in this bioarchive paper. And indeed, he does see a decreased exosome, what he called exosome secretion, yes. So that has been done. And for the tagging, yeah, I think tagging to synthetically load the molecule into exosome using the CD63 tag, yeah, that would be really interesting. What we think we have here now is a way to distinguish better what is exosome and ectosome, and thus then subsequently be able to use that to discriminate much better what the functions of the different EVs could be. But yeah, I just wanted not to mention Steve Gould, not to make people think that we don't want to quote his work because we have the same results. We just don't call the vesicles the same way, but we have a similar result.
Brilliant, thank you.
So then about response to stimuli, yeah, it might be interesting. I don't know if you thought about specific environmental change that might be tried for this. Because I don't know for ELA to what they could answer, except maybe stress or something. Then in a more complex environment, yes, it would be interesting also to look at this. I don't know for imaging if we want to do the same kind of live imaging. I don't know how feasible it is to, and if it would be that clear to image something in 3D. I guess it's feasible, but I don't know much about this. And then for the next steps, so one interesting thing would be to identify some genes that are involved specifically to the secretion either of ectosomes or endosomes. So for this, I started to do siRNA experiments to look at some proteins that might affect either the secretion of CD63, CD9 or the other markers into it. But so yet we didn't have so very conclusive results, let's say. But it's something that would be interesting to look at. What we is how we can to identify some mechanisms of secretion and genes that are specific of the secretion of either exosomes or And then about the apoptotic bodies, then I guess we could try to induce apoptosis and check what we have and how it changes the observations that we made. The only problem would be how we differentiate if apoptosis itself induce, because if you have also small apoptotic bodies that are co-isolated with the classical EVs, let's say, then we should have a way to discriminate if the apoptosis increases the EVs that we observe here, so the exosomes and ectosomes themselves, and in addition, some small apoptotic bodies. So we might need really markers that allow to discriminate this, but it's also something that is interesting, yes, indeed.
I think then as well, there are some, thank you for the answers. And it looks like some questions in the chat box.
We have some questions in the chat box. So Laura has a question. Could you go ahead and I can unmute you and you can go ahead and ask your question.
Hi, can you hear me?
Yes, we can hear you.
Different computer. Super beautiful paper, and Erin, you did a great job explaining everything. I recently found, I've been doing some work on Rab proteins, and there was a paper recently published that Rab31 is involved in CDCC3 trafficking to the multivesicular body. So I was wondering if you found any, or looked at any differences in Rab proteins, both between ectosome versus exosome, or with time, because I saw plasma membrane had higher, or not, maybe not secretion, but higher amounts at 24 hours versus lysosome at 3. Sorry, the proteomics slide went a little quick. But it was no, it's fine. I can be a little slow. But it was really interesting. So I just was wondering if you saw any differences in Rab proteins.
In terms of Rabs that are found in the EVs, in the proteomic, or the role of the Rabs into the secretion?
Rabs in the EVs. Because Rab proteins can be secreted. Well, I found that Rab proteins can be secreted, but different cell lines, I'm not 100% sure.
So in the proteomic, I think we didn't notice anything particular. Actually, we didn't find so many Rabs we didn't identify so many Rab proteins. Well, no, we didn't see anything particular.
Yeah, thanks. I just wasn't sure.
So for the next question, we have Elisa. Could you go ahead and ask your question?
Yes, thank you. Wonderful presentation. The paper is very nice. I have a bit of a technical question about the ultracentrifugation. I'm a bit confused on how the speed is determined, because we know we have the minimum, the average, the maximum radius of the ultracentrifuge, and I'm not sure if there is that everyone is doing the same thing. So I was just curious to see how you do the calculation and what's the principle behind it. Yeah.
Well, the speed that we use, I think it's quite classical. That's something you can find in many papers. And we check just based on each cell line. We just verify that what we isolate is a correct amount with the expected markers. But then for the calculation, it depends just on the rotor that you use and the machine that you use. I guess that you can always do the calculation.
Yeah, no, but I mean that for what I know, in general, you need to calculate the G, the speed, according to where your vesicles are supposed to go. That's the principle I've been using. So if I'm using a sucrose gradient, I know that the vesicles will most likely localize in the middle of my vial. So we use the average speed to do the RPM to G calculation. But I'm not sure if this is done instead when you use media or other things, because it actually changes things a lot. You think you are centrifuging at 10K, but if you do the calculation wrong, maybe you're actually centrifuging at 30K as maximum radius. So that's why I'm asking. I know it's a bit of a technical question, and I don't know if there is any agreement on it, but I was not able to find any paper that explains this. So that's why I'm asking.
No, so I know there's, I don't know if this helps, and I'll post the link in the chat box. But there was a paper, I think it's Jan Lotval's, and they kind of look at the ways of standardizing, because of the different rotors and things that you can use, trying to standardize that across the field. But obviously it's still something that we've got a way to go with. But and in the Mathilde's paper and Clotilde's paper, they make it clear which rotors they're using. So then you've got those measurements are available if you need.
Yeah, I just don't think it's a matter of rotor. It's just the principle that you use independently from the rotor. But I don't want to take too much time on this technical question if you don't have a specific answer.
Well, just the answer here, we use the maximal, not the average, but the maximal distance to calculate, because we just want to have we know the vesicles will go at the bottom of the tube.
Yeah, okay. So that's exactly what I wanted to know. That's the principle that I was using too, but I was not sure. Thank you.
So our next question is from Esther. Could you go ahead and ask your question?
Yes, thank you very much. First of all, very nice, Mathilde, to see that you now use this technology to really look at EV secretion and EV subpopulations, and well done, Aaron, on presenting this work. My question is about the proteomics part, because I was a bit surprised to see, especially at the 3-hour time point, that the CD63 EV population is very much enriched in ER proteins rather than endosomal proteins. So yeah, what kind of ER proteins do you see there, and do you have any thoughts on this result?
So what kind of proteins exactly? I should check on the list. I don't have them in mind now, sorry. But yeah, I don't know exactly why. It's not something we looked at in details. But maybe also one thing to keep in mind is that at 3 hours, the material that we have is very low. And maybe we have also more chances there to have some contaminants that are more abundant compared to all the other proteins. So it might be this, or it might be specific. This I don't know. But yeah.
Okay. Maybe another question, just from a curiosity viewpoint. Many HeLa cells that people culture in labs are quite different. Did you see the same kind of results in different HeLa lines that you use, or you use only one HeLa line?
So in the paper, I use always the same line for now, but it's true that now we have started to think about also comparing with some HeLa that we know are different. So that's something like maybe we will try to do in the close future.
Thank you.
Now we have Mandy Chan, who has a question about quantitation. Could you go ahead and ask your question?
Thank you for the great presentation too. Have you used any quantitation technologies such as things like NanoView to confirm co-localization on the released single EV? And have you compared the abundance of the exosome versus ectosome that you find? Which one do you think is more abundant? This is like similar to the work that is done by Stephen Gould, and they use NanoView extensively to do study co-localization. Yeah, I'm interested in the relative abundance and co-localization.
Yes, so yes, I had sent some samples to NanoView actually, yes, to check on this. And indeed, I saw also this co-localization of some EVs of both CD63 and CD9, and some EVs that have only one of them. Then about the relative abundance, so we didn't know if really we should trust completely these results or not. So, and we wanted also to compare with other techniques, but so far we didn't end up to have one technique that we would sure to have a really reliable result. So I don't want to say something now that might be wrong later, because we didn't get so far to this. But that's something also we considered doing, yes. And for the results, from the results that we have until now, so we think that for HeLa, at least we have more ectosomes that are secreted than exosomes. But in terms of quantity and precise percentages, I cannot answer precisely.
Thank you.
Our next question is from Ganesh. You can go ahead and ask your question.
Hey, hi, Aaron. Nice summary of the talk and great work here. I have a quick question regarding the data that you come from the Rush construct. So if you block the transfer of proteins from Golgi to ER and different compartments, so if you use inhibitors like monensin or some other inhibitors that inhibit the ER to Golgi trafficking, do you think if accumulating those compartments would enhance the trafficking of those compartments to the MVB or lysosomes that access? And will that affect the release of more CD9, CD63 positive EVs together?
I don't know this. Why you think that if you block a protein from going to the Golgi, it might go directly to MVBs?
So basically you're not letting them exit out. So the cell might want to recycle them or degrade them. And during that process, whether this MVB formation would happen or not. So that was my question. It's just trafficking.
Okay, yeah, I don't know this actually, but yeah, why not? It's something might be interesting to look at. Yeah, I don't know if the protein would just remain trapped in the ER in this case, or might go, yes, in endosomes. No, I don't know, but yeah, that's interesting.
Thanks.
Thanks.
Then we have a question from Rashgul Alam. Could you go ahead and ask your question?
Thank you. Thank you for a nice presentation. Actually, I have one question about ear calcium. When the ear stress is increased, that time the ear calcium is released from the ear, and the cytosolic calcium is increased that time. So it's how to regulate the exosome release from cell. There have any inhibition of because I have another question. In that times the ear stress is increased, that times is lysosomal calcium overload also increases. So it possible to increase the ear from the cell release from exosome release from the cell.
Well, I know that you have some regulations involving calcium and calcium channels that are shown to regulate EV secretion. And specifically about calcium that is in the ER, I don't know actually. No.
Actually, I saw different article in research article I saw. During the ear calcium is released from the ear to cytosol. That times is stimulate the exosome release from cell. But my question is, during the ear stress, that times also the calcium overload into the lysosome. So this is our lab findings during the ear stress. That times is calcium is overload in lysosome. So lysosomal rupture is happened that times.
Okay. Well, yeah, no, I have no idea about this, but that's yes, if it's what you found, it's interesting. So you say that it's when you have a stress in the cell, then the ER releases calcium. Okay. No, I don't know. I have no clue, but. Yeah.
And another question is technical question. I use the TFF, but after the TFF filtration and left of the media is remain. That means minimum 5 ml. But I did not isolate the exosome from this 5 ml. How can I can isolate my exosome from this TFF?
TFF, what is it? Sorry.
TFF is the.
I think it's tangential flow filtration. Is that?
Yeah. Transient flow filtration.
So basically, you concentrate your media with this technique, and then you want to further isolate.
Actually, I culture the cell, then I make ear stress in cell. Ear stress inducer, I use the tafsigarzine, and because our lab findings also, I also mentioned that calcium during the ear stress inducer use, that times this calcium is more increases in cytosol. So our findings is if the cytosolic calcium is increased, that times the exosome is more released from the cell. So we use the media in the TFF. That times the 5 ml of media remain after the TFF. So, but I did not isolate the exosome from this 5 ml. I have to ultracentrifugation that times from the 5 ml media.
So we are sorry, but we are not using TFF in my group, so I'm afraid Mathilde cannot answer. You know, people who use TFF a lot is Bernd Giebel or Eva Rode in Germany, so in Germany and in Austria. Probably many people in the US also, so I guess we cannot answer this question because we are not using the TFF. Sorry about that.
So now we have another question from Nico Hutman. You can go ahead and ask your question.
So my question was regarding the 200K isolation. So I guess you compared a second subsequent 100K isolation from the same supernatant. And my question is if it was entirely cleared of EVs so that you isolate a new, well, let's say population fraction of EVs with the 200K, or if it's just the residual EVs, which have not been cleared by the first step. So that was my question.
No, I didn't check that in so much detail, I have to say. Let me think. No, I don't know. The only thing I did was to do an EM on the 200K, but which corresponds to the 100K plus 200K directly done together. And okay. I see EVs from the smallest that I can observe are 45 nanometers, but I have to say that I didn't compare to 100K alone. So yeah, something that might be done, but I didn't do it.
Okay, great. Thank you.
So I don't see any more questions here in the chat box. So, well, I can send out a poll earlier about how they pronounce the abbreviation of the student network for EV. So we'll end this poll and we can share with you how to actually pronounce student network on EV. So we have 56% voting for SNEV. So I guess we're going with SNEV. And this is, I think 85 people answered this poll. And so if no one has any other questions, I would just want to thank again Aaron for that clear presentation. And thank you, Mathilde and Clotilde, for helping out with the discussions. And also thank you again for letting SNEV co-host this journal club.
Well, I want to thank Aaron.
Go ahead.
Sorry, I just wanted to say thank you very much, Aaron, for really presenting the work in a very clear and very thorough manner. Thank you.
No worries.
Yeah, thanks, Aaron.
It was good fun.
All right, well, and thanks to all of our attendees as well. For those of you who answered questions, also took part in the poll. And those of you who are in the minority on SNEV, you know, you'll still have an opportunity to participate in SNEV events. So no worries there. So thank you all. Have a great rest of the day, and we look forward to seeing you again soon. Bye now.